Hey everybody, and welcome back to Non-Mendelian Genetics 2. So we're going to pick up talking about another one of the ones that doesn't follow Mendel's basic rules, and this is sex chromosomes. And so with sex chromosomes, we have sex-linked genes. Thomas Hunt Morgan experimented with fruit flies, and that's where this is where the idea of sex-linked genes really came out. That they discovered this and determined that specific genes can be carried on the sex chromosomes. We know a lot more about that now, so sex-linked genes are going to be located on either the X or the Y chromosome. Y-linked genes are found specifically on the Y chromosome, and only males will ever get the Y-linked traits because only males have Ys as one of their chromosomes. That the Y is what makes them a male. There are very few Y-linked genes. First of all, because the Y is very small. Secondly, so we just don't have a lot of disorders. And like I said, these are just only found in males. X-linked genes, however, are going to be found on the X chromosome. And so they can show up in a female if she gets 2 copies, or in a male, it only takes 1 because they only have 1 X chromosome. So when we're talking about inheriting these X-linked genes, that's what we focus on. Fathers can pass an X-linked allele to all of their daughters, but none of their sons, because a father will send a Y chromosome to them. Mothers can pass X-linked alleles to both daughters and sons. So if a capital N stands for normal and a little n stands for affected, you will place these on the sex genes, so X or Y. Notice that because this is an X-linked trait, it only shows up on the X's, not on the Y. So we have a normal mother and we have an affected father. When we do this cross, we find that all of the males are normal, all of the females are normal, but they carry the affected trait and then can pass it on to their children. So if we have an affected mother, meaning notice she's carrying the affected trait, she can pass it on to sons, and she can also have carrier daughters. If an X-linked trait is due to a recessive allele, females will only express the trait if they are homozygous for that affected recessive allele. Because males only have 1 X chromosome, they will express the trait they inherit from their mother. Period. End of story. When it comes to X-linked traits, males always get from their mothers. These specifically are called hemizygous, because heterozygous doesn't really apply since you only have 1 copy, and heterozygous is saying you have a mixture of 1 of each types. So due to this, males are more likely to have an X-linked disorder since they don't have a way to cover it up with another. So Duchenne muscular dystrophy is a progressive weakening of muscles, hemophilia, inability to properly clot, color blindness, inability to correctly see colors. All of these things are X-linked disorders. Now here comes the really crazy twist when it comes to the X's. We call this X inactivation. So we know that females inherit 2 X chromosomes, twice what males have. So what happens is during development, most of the X chromosome in each cell becomes inactive. The inactive X in each cell of a female condenses into what we call a bar body. This helps to kind of regulate the gene dosage in females, because if you think about it, females are getting twice as much of the gene as males. So if you can basically inactivate one copy of it, then the gene is going to kind of run at a normal level or normal rate. And then we have linked genes. So with linked genes, we bring in this concept of genetic recombination because this is really important to understand when we're talking about linked genes. So when we're talking about genetic recombination, what we're saying is the production of offspring with a new combination of genes from parents. So we're mixing up the genes. Parental types, offspring with the parental phenotype. The recombinants are offspring that have phenotypes that are different from the parents. Okay. So Mendel observed recombinants during his crosses. For example, he saw a green wrinkled plant crossed with a yellow round plant. And so 50% were the parental phenotypes and 50% were recombinant phenotypes. So he did actually observe this concept. 50% recombination indicates that genes are unlinked or on different chromosomes. So when we see this 50% like this, what we're saying is that they are totally separated. They are not in any way connected. But the problem with linked genes is they're located really near each other on the same chromosome. So they tend to be inherited together. They don't really get separated out when we're doing the inheritance process because they're so close to each other on the same chromosome, that whole chromosome goes. So meiosis and random fertilization generate genetic variation due to the independent assortment of the chromosomes, crossing over in meiosis 1, and of course the fact that any sperm can fertilize any egg. With linked genes, we see the parental phenotypes in offspring at a higher than 50% rate because they're so close together that they typically get transferred together. But during crossing over, chromosomes from a paternal chromatid and a maternal one can exchange segments. So it is still possible that they will get separated out, but it's less likely. So that's why we see a higher than 50% rate for the parental phenotypes. So crossing over helps explain why some linked genes get separated during meiosis. And we found that the further apart the two genes are on the same chromosome, the higher the probability that crossing over will happen. Therefore, the higher the recombination frequency. So the closer they are together, the less likely they're going to recombine. So this takes us to this concept of mapping distance, because obviously if we're talking about how far apart are these things, can we map it? So there were experiments performed by Sturtevant that allowed scientists to start mapping genes and their locations on the chromosomes. And we've done a lot of this now with the human genome. So we form a linkage map. So it's a genetic map that is based on recombination frequencies. The distance between the genes are what we call map units. One map unit is equivalent to a 1% recombination frequency. So the more map units, the higher the recombination frequency will be. This is the way we express the relative distances along the chromosome. 50% recombination means that the genes are far apart on the same chromosome or on two different chromosomes, because they're more likely to be separated and rearranged. So here's a mapping distance. You see our map, and the different numbers represent the distance from, in this case, the ariste trait. Okay. And so here's our ariste at 0, and then the further we get from that, we see a higher distance. And then, of course, we have the non-nuclear DNA. So we know that some traits are actually, the information is located on the DNA that's in the mitochondria or in the chloroplast. Both chloroplasts and mitochondria are randomly assorted to gametes and daughter cells. So the ones that actually get placed into any egg or sperm is just kind of a random, here, we're going to shove this one here, this one goes over here. So we still get that kind of randomness, that variation. In animals, mitochondria are transmitted by the egg, not the sperm. All that's transmitted by the sperm in humans and in animals is the DNA. Nothing else. The egg has all of the other organelles, so they all come from the maternal line. So all mitochondrial DNA is maternally inherited, and they can actually trace people's lineages and connections even between different families through the mitochondrial DNA through that maternal line. So theoretically, could you say, well, then all of the mitochondrial DNA should be the same, because if all organisms started with one specific individual, but remember, there is mutation. So you're going to see various maternal lines. In plants, mitochondria and chloroplasts are transmitted in the ovule, not the pollen. So once again, these are transmitted via the maternal or female line of the organism. So mitochondrial and chloroplasts that are determining traits are maternally inherited in plants, in animals, all of these things. Okay, so hopefully that gives you a little bit of information about our non-Mendelian genetics, and we'll see you for the next lesson. Bye.